Okay, so I just wanted to do a very quick run through of how you would analyze your sequences from Sanger using DNA Subway so we can actually view our electropherogram here. So if you search for DNA Subway, we're using the dnasubway.cyverse.org. And if we go here, what we're going to do is we're actually going to enter as guest. You can make an account if you want to, but you don't need to. And we're going to look for determine sequence relationships. That's the blue line. Okay, once you press on that, we're going to select that we want to do. We're targeting the 16S gene. That's what we sequenced with our PCR product and everything. So we select for 16S, and then we're going to upload our AB1 trace file, which you guys have access to now. And I actually downloaded one just as an example, and I'm going to drag and drop it into here. You can also choose to upload the files directly from your downloads. You have to give it a project title, and so for this one, let's just say I'm doing student 24, and then we press continue. So it runs really fast if you're only doing one sample, but you can do up to like 40 samples at once if you wanted to see what your classmates got. And so when we go and press on Sequence Viewer, we can actually copy paste this right here. So we'll just select it all and then copy paste into a Google Doc or any sort of like Word doc, something that you can edit in here. And I'm just going to put in here student 24. So I know that this is the name of my sequence. Okay, we're going to go back to DNA Subway and we're going to press on this little graph icon. So here we can now see our electropherogram, and you can actually see how the bases are calling for each individual base pair all the way through. So what we're going to do is we're going to trim this so that we only have high quality sequence data that's used in our BLAST search. So all of this junk over here, we're going to trim it. So we're going to trim probably until, for this sequence, it's pretty high quality. So we'll trim right around 53 off of the front. So we can go over here and we can go and turn on word count. So if we do word count, display word count typing, and then down the bottom, just make sure it's characters that are selected. And so I believe I said 52. So we're going to go and look for about 52. And then we're going to trim that off. So we just delete it. Go back to DNA Subway. And then we're going to look at the tail end of this. And when the sample starts to become lower quality, that's when you'll start to see bases overlapping like this. And so that's when it's like unsure of what is really the proper base. And you can see that the sample starts to look a little bit different near the tail end. We're also going to throw that out. So it ends at 870. And we should probably cut it around, probably around 770, maybe 780, where you can start to see there's not like too much overlap. But definitely by 790, things are starting to have extra peaks. So maybe for this one, we'll cut off at about the 780 mark. Okay, so 780, 870, so we want to cut off 90 from the end. Okay, so we're going to take off 90. From here, this is now called a FASTA file. Okay, so this is a FASTA sequence. And so what we can do here is we can copy this. If you want to give it a name, just always make sure that you have the little caret sign in front of it here, because otherwise it will mess up the BLAST search. But so now that you have your FASTA sequence, you can go and search for BLAST NCBI. And once you are in BLAST, you press the nucleotide BLAST, and then we're going to paste our FASTA file that we, our FASTA sequence that we made. Okay, from there, everything is already populated to the ones that we want. So we'll just press BLAST. And this can take a couple seconds, a couple minutes to run. It just depends on how many people are using BLAST at the same time as you are, or how many sequences you're running, because you can run multiple sequences at one time. They just need to have their own name. And so you can see for this one here, we have the main top hits are all bacillus. So it's very likely that this particular bacteria is a bacillus. So in terms of the query coverage, we had 100% coverage. So that means our full input was covered. It matches entirely in its length to something that's already populated in BLAST. And it's very, very similar. It's 100%. So with high certainty, it's a bacillus species. And maybe it's Bacillus subtilis, but it could also just be a general Bacillus species, although the top hits all do seem to be Bacillus subtilis. So if you go to graphic summary, you can see based off of our query length itself was 728 base pairs. And so that's this teal line up here. And so it shows you, these are all the BLAST searches that the hits that we got on the database. Okay, so all those red lines, they stand for each one of these. And so based off of that, you can see everything's lined up fully with the query. You can also go into the individual alignments to see where things are lining up. So the query, which again is our FASTA sequence, is going to be the top line, and the subject is what it's matching up to in the NCBI database. And so for this one, it's 100% match, as we saw earlier, and you can also see here in the percent identity, it's 100% match. So every single one of the lines are showing that they're the same, but if we were to scroll down, maybe we'll find one that has a break in the matches. So it's not 100% identity, but instead is like 99% or 97%. This bacillus seems to have a lot of hits inside of NCBI, which makes sense because Bacillus subtilis and bacilli in general are very common bacteria. But your particular isolate, your unknown, might be something that's different and not as well known. So if it has less than a 95% identity match, then that means that it's not going to be of the same species for sure, and maybe you are only confident in the genus. Okay, so you are going to need to include this in your lab report, and we want you to record all of these values in your lab report, and just to make sure that you understand how to do this process and see what exactly your bacteria might be. So if you guys have any questions, feel free to talk to any of your TAs and or just bring it up during class next time we meet.